Setter.
I am Arina binti Abdul Rashid.
I'm Nurhasliza binti Muhammad Nur.
So today we are going to present our lab assignment, which is the development of tropical fruit, especially in banana. Why we choose banana is because we can easily find banana here in Tanjung Malim, and it is cheap, and we also love banana.
For your information, banana is the fruit of genus Musa of a family of Musaceae. Banana are giant monocotyledonous perennial herbs. After banana plant produce seed fruit, it dies. Have you ever heard of Malaysian idioms? Pisang tidak akan berbuah dua kali. Next, banana is a tropical plant which grow best under warm condition.
Next, this is the overview about the banana tree. First, banana tree have underground comb and trunk, which is known as pseudostem. You can see here, this is the pseudostem. Banana tree looks like a tree, but it's not a tree, it's a herb perennial. Then banana has terminal inflorescence, which are the plants where fruit produced. Then the leaves of banana is broad, long and graceful, and it has adventitious fruits.
the flower morphology of banana. As we can see here, this purple one is the brake. And when the brake is pulling from the banana heart, it will causes the brake scar. This is the rachis. Next is the banana heart. The inflorescence is a complex structure that includes the flowers that will develop into fruits. This inflorescence, which the main axis continues to grow, and the lateral branches have determinate growth. The brake is a modified leaf, which associated with a reproductive structure, such as a flower. The brake exposes a cluster of male flower, and the male flower or the staminate flower each subtended by a brake. In figure 1.1, we can see that the male flower can be found inside the brake. This is a closer look of male flower. In figure 1.2, the upper part of the male flower is the ovary, and the globular shape is the stigma, and this is the loop of compound tepal. Next is the tepal. The sepal and petals of banana cannot be differentiated, therefore it was called tepal. And this is the tepal of the banana. Figure 1.5 shows the structure of male flower. As we can see here, the male flower has slender stele, and it consists of 5 stamens with long anther. This is the anther. The stigma located at the tip of stele. The male flower has smaller ovary, and its ovary is not functioning. The male flower ovary shape is arched and the color is white. Figure 1.7 shows the stigma, the stele, and anther, and this is the loop of compound tepals under dissecting microscope.
Next, I will present the reproductive system. As you can see here, male, which is known as stamen, has anther and filament. Meanwhile, the female part, which is known as carpel, has stigma, stele, and ovary. First is stamen. Stamen is a male reproductive organ.
For stamen of banana flower, it have club-shaped structure which is cylindrical and enlarging gradually towards the end. Then it have 5 stamens, long anther. Figure 2.0 shows the anther and the filament.
On the anther, there are many pollen grains.
The pollen grain located at the both sides of stamen, and then the color is white, which is recorded at the line where the anther split to release the pollen.
Next, we move to the female reproductive organs.
For the stele basic color is white and the shape is curved at the base. Then for the stigma, it is the sticky knob at the top of pistil and the color is cream. Stigma of banana flower have sticky substance in which are adapted to trap the pollen to initiate the fertilization. Lastly, the stigma is the part of pistil where pollen will germinate. This is the closer look of the stigma. You can see the sticky substance at the knob of stigma.
Second is ovary.
For the banana ovary has 3 locules and the placentation is axial. But on the slide we can't see clearly because the slide is already oxidized. Each locule have 2 rows of ovule. You can see on the figure 2.5 which shows the ovule of the banana. This is also the cross-section of ovary.
Next, we are going to talk about pollination in banana flowers.
For a plant to be self-fruitful, it sets fruit from its own pollen rather than needing to be pollinated by a separate plant or different variety. Banana produce fruit without pollination. The fruits develop without pollination at all we call as parthenocarpy, which is from the female flowers. Thus, the seeds produced are sterile and will not germinate.
Next, I'm going to tell you guys about pollen germination.
After we ran the experiment for 2 days, a long pollen tube was formed. Right here. We germinate in a medium consists of salt and sucrose solution. The sugar in the medium acts as osmotic regulant, which regulates the diffusion rate of water from the medium to pollen grain.
Next, I'm going to tell you guys about fertilization, which include endosperm and embryo development.
The development of endosperm in Musa is the nucleotide. In nucleotide of endosperm, the first division of primary endosperm nucleus and a few subsequent nucleus divisions are not accompanied by wall formation. The nuclei produced are free in the cytoplasm of the embryo sac, and they remain free indefinitely, or wall formation takes place later. In Musa, not only nuclear division is non-synchronized, but some nuclei divide more actively, forming isolated vesicles or nodules that develop into separate endosperm masses. The seed has two chambers. The larger one contains the endosperm and the embryo, while the smaller one contains the callosal mass. After germination, the process followed by embryo expansion along its axis through cell elongation, and ends with the breaking through of the seed coat by the embryo root and shoot. In the banana seed, the embryo disappeared within 2 weeks or 3 weeks. The first evidence of embryo germination occurs when microphylla plugs grow from microphylla canal. According to Simon, the growth of microphylla plug is caused by the pressure produced from the embryo growth, as well as the pressure resulting from the absorption of water of its environment. Studies using ruptured seed coat shows that the expanding embryo emerged through the rupture, the path of least resistance, that some force is needed to dislodge the microphylla plug.
Next, we are going to talk about seed production and its development. How seed produced in banana?
Banana are formed without pollination, so the seeds are sterile and will not germinate. To produce new plant, they must propagate vegetatively. The tiny black dot in the center of banana are the remnant of ovules that won't become viable. The seed filled with a conspicuous white and have a large amount of powdery endosperm. It have linear embryos, and banana seed have large, thick, and hard testa.
Last but not least, we're going to talk about fruit production and its development. Okay, how banana fruits produce?
Banana fruit develop from banana heart in a large hanging cluster made up of tears. These fruit are long in shape with yellow and green skin, like we all know. The outer protective layer of each fruit is known as skin or peel. Is a fusion of hypanthium, which is floral receptacle, and outer layer of the pericarp. The endocarp is a thin layer, while the mesocarp is the major edible part of the fruit. During the development of the fruit from the ovary, the tepals, stele, and staminode abscess, leaving a characteristic colored scar at the tip of the fruit. Edible pulp, which is a starchy parenchyma which fills the fruit in parthenocarpic types and surround the seed in seeded bananas, mostly originates from the outer lining of the locules.
That's all from us. Thank you. Have a nice day.